Digestion and absorption of carbohydrates. Now we are going to describe the general principles of digestion and absorption, describe the steps involved in the breakdown and absorption of carbohydrates, explain absorption of water, electrolytes and minerals in the intestine. First term is the condensation, the process where two monosaccharides are joined together with the removal of hydrogen and one hydroxyl ion from each. Then dimers formed when two small identical or similar molecules called monomers are linked together. Then hydrolysis, a process of returning the hydrogen and hydroxyl groups from water to the polysaccharides and thereby separating them with the help of enzymes. Monomers, molecules that can combine with other similar kinds to form a polymer. Then oligomers. Oligomers are molecules that consist of a relatively small and specifiable number of monomers that are usually less than 5 in number. General principles of digestion. The major foods on which the body lives, with the exception of small quantities of substances such as vitamins and minerals, can be classified as carbohydrates, fats, and proteins. They generally cannot be absorbed in their natural forms through the gastrointestinal mucosa, and for this reason they are useless as nutrients without preliminary digestion. Digestion is a process that is characterized by a specific sequence of events following the ingestion of foods. These events allow the foods to interact with the various secretions such as enzymes, emulsifying agents, acid or alkaline substances, thereby facilitating the breakdown of complex molecules into simpler molecules under optimum pH. The various events associated with the process of digestion include the following: mixing and lubricating the food with secretions of the GI tract, ensure uniform homogenization, enzymatic secretion from various glands and cells, lining the GI tracts, break down complex molecules into simpler molecules such as oligomers, dimers, and monomers. All digestive enzymes act by hydrolysis. Most GI tract enzymes are secreted as inactive precursors which are then activated in the GI tract. Secretion of acid or bicarbonate from the GI tract ensures optimal pH for digestion. In the case of fats, secretion of emulsifying fats such as bile acids help in emulsifying dietary fat, thereby promoting fat digestion. Final digestion of most oligomers and dimers occur at the small intestine brush border, resulting in release of monomers that are finally absorbed. Therefore, this chapter discusses the processes by which carbohydrates are digested into small enough compounds for absorption and the mechanism by which the digestive and end products as well as water, electrolytes and other substances are absorbed. So, digest of the various foods by hydrolysis. Hydrolysis of carbohydrates. Almost all the carbohydrates of the diet are either large polysaccharides or disaccharides, which are combinations of monosaccharides bound to one another by condensation reactions. This phenomenon means that a hydrogen ion has been removed from one of the monosaccharides and a hydroxyl ion has been removed from the next one. The two monosaccharides then combine with each other at the sites of removal, and the hydrogen and hydroxyl ions combine to form water. When carbohydrates are digested, this process is reversed and the carbohydrates are converted into monosaccharides. Specific enzymes in the digestive juices of the GI tract return the hydrogen and hydroxyl ions from water to the polysaccharides and thereby separate the monosaccharides from each other. This process called hydrolysis is the following, in which R double dash R dash is a disaccharide, where R double dash R dash plus H2O, digestive enzyme ROH plus RH. Digestion of carbohydrates. Carbohydrate foods of the diet. Only 3 major sources of carbohydrate exist in the normal human diet. They are the following: sucrose, which is the disaccharide known popularly as cane sugar. Lactose, which is a disaccharide found in milk. Starches, which are large polysaccharides present in almost all known animal foods, particularly in potatoes and different types of grains. Other carbohydrates ingested to a slight extent, which are amylose, glycogen, alcohol, lactic acid, pyruvic acid, pectins, dextrin, and minor quantities of carbohydrate derivatives in meats. The diets also contain a large amount of cellulose, which is a carbohydrate. However, no enzyme capable of hydrolyzing cellulose are secreted in the human digestive tract. Consequently, cellulose cannot be considered a food for humans. Digestion of carbohydrates begins in the mouth and stomach. When food is chewed, it is mixed with saliva, which contains the digestive enzyme ptyalin and alpha amylase secreted mainly by the parotid glands. This enzyme hydrolyzes starch into the disaccharide maltose and other small polymers of glucose that contain 3 to 9 glucose molecules. However, the food remains in the mouth only for a short time, so probably not more than 5% of all the starches will have become hydrolyzed by the time of food is swallowed. Starch digestion sometimes continues in the body and fundus of the stomach for as long as 1 hour before the food become mixed with the stomach secretions. Activity of the salivary amylase is then blocked by the acids of the gastric secretions because the amylase is essentially inactive as an enzyme once the pH of the medium falls below about 4.0. Nevertheless, on average, before food and its accompanying saliva becomes completely mixed with the gastric secretions, as much as 30 to 40% of the starches will have been hydrolyzed mainly to form maltose. Then digestion of carbohydrate in the small intestine. Digestion by pancreatic amylase. Pancreatic secretion, like saliva, contains a large quantity of alpha amylase that is almost identical in its function to the alpha amylase of saliva but is several times as powerful. Therefore, within 15 to 30 minutes after the chyme empties from the stomach into the duodenum and mixes with pancreatic juice, virtually all the carbohydrates will have become digested. In general, the carbohydrates are almost totally converted into maltose and or other small glucose polymers before passing beyond the duodenum or upper jejunum. Hydrolysis of disaccharides and small glucose polymers into monosaccharides by intestinal epithelial enzymes. The enterocytes lining the villi of the small intestine contain 4 enzymes: lactase, sucrase, maltase, and alpha dextrinase, which are capable of splitting the carbohydrates lactose, sucrose, and maltose, plus other small glucose polymers into their constituent monosaccharides. The enzymes are located in the enterocytes, converting the intestinal microvilli brush border, so the disaccharides are digested as they come in contact with those enterocytes. Lactose splits into a molecule of galactose and a molecule of glucose. Sucrose splits into a molecule of fructose and a molecule of glucose. Maltose and all other small glucose polymers split into multiple molecules of glucose. Thus, the final products of carbohydrate digestion are all monosaccharides. They are all water soluble and are absorbed immediately into the portal blood. Basic principles of gastrointestinal absorption. It is suggested that the reader review the basic principles of transport of substances through the cell membrane. The following paragraphs present specialized applications of this transport process during GI absorption. So, anatomical basis of absorption. The total quantity of fluid that must be absorbed each day by the intestine is equal to the ingested fluid, about 1.5 liter, plus that secreted in the various GI secretions, about 7 liter, which comes a total of 8 to 9 liter. All but about 1.5 liter of this fluid is absorbed in the small intestine, leaving only 1.5 liter to pass through the ileocecal valve into the colon each day. The stomach is a poor absorptive area of the GI tract because it lacks the typical villus type of absorptive mechanism and membrane, and also because the junctions between the epithelial cells are tight junctions. Only a few highly lipid soluble substances such as alcohol and some drugs such as aspirin can be absorbed in small quantities. Folds of occurrence villi and microvilli increases the mucosal absorption area by nearly 1000-fold. Many folds called valvulae connivence or folds of Kerkring, which increase the surface area of the absorptive mucosa about threefold. Also located on the epithelial surface of the small intestine all the way down to the ileocecal valve are millions of small villi. These villi project about 1 mm from the surface of the mucosa and the presence of villi on the mucosal surface enhances the total absorptive area another 10 to 10-fold. Finally, each intestinal epithelial cell on each villus is characterized by a brush border consisting of as many as 1000 microvilli that are 101 μm in length and 0.1 μm in diameter and protrude into the intestinal chyme. These microvilli are visible under the electron microscope. This brush border increases the surface area exposed to the intestinal material at least another 20-fold. Thus, the combination of the folds of Kerkling, the villi, and the microvilli increases the total absorptive area of the mucosa perhaps 1000-fold, making a tremendous total area of 250 or more square meters for the entire small intestine, about the surface area of a tennis court. Then the advantageous arrangement of the vascular system for absorption of fluid and dissolved material into the portal system, and the arrangement of the central lacteal lymph vessel for absorption into the lymph. There is a villus and many small pinocytic vesicles which are pinched off portions of enfolded enterocyte membrane forming vesicles of absorbed fluids that have been entrapped. Small amounts of substances are absorbed by this physical process of pinocytosis, extending from the epithelial cell body into each microvillus of the brush border are multiple actin filaments that contract rhythmically to cause continual movement of the microvilli, keeping them constantly exposed to new quantities of intestinal fluid. Absorption in the small intestine. Absorption from the small intestine each day consists of several hundred grams of carbohydrates, 100 or more grams of fat, 50 to 100 grams of amino acids, 50 to 100 grams of ions, and 7 to 8 liters of water. The absorptive capacity of the normal small intestine is far greater than this, each day as much as several kilograms of carbohydrates, 500 grams of fat, 500 to 700 grams of proteins, and 20 or more liters of water can be absorbed. The large intestine can absorb still more water and ions, although it can absorb very few nutrients. Mouth. When we just look at the overview of this digestion of carbohydrates, digestion and absorption of carbohydrates in the GI tract, GI gastrointestinal tract, we can see in the mouth what is happening. There is an enzyme called alpha amylase. Tyalin. Parotid glands secrete them and acts on starches to form maltose and glucose 3 to 9 polymers. Then esophagus is there. Stomach contain enzyme, another enzyme similar to the same enzyme, alpha amylase or tyalin, which is secreted by the parotid glands. Then digestion continues in the stomach for up to 1 hour. 30 to 40% starch is converted to maltose and glucose polymers. Then after that, it reaches small intestine where alpha amylase from pancreas is there. Then 50 to 80% starch is converted to maltose and glucose polymers before passing beyond duodenum or upper jejunum. Then large intestine is there. There is in the small intestine there is another enzyme, lactase, sucrase, maltase, and alpha dextrinase enterocytes lining the villi. Lactose convert lactase lactose is converted into galactose and glucose with the help of lactase, and sucrase converts sucrose into fructose and glucose, and maltase converts maltose into glucose and another molecule of glucose. 3 to 9 glucose polymers are converted into glucose like alpha dextrinases. Salivary amylase, tylenol is there, and D-xylulose, lactose, sucrose, starch, maltose, etc. are the substrates, and they enter into the stomach region, and there will be pancreatic amylase, and that is going to react with these food items. Then maltose, sucrose and lactose secretion and cholecystokinin reaches the intestinal wall by vagus nerve is also connected with that. Portal vein is also has connections. Epithelial cells are there. Then galactose, fructose, glucose, etc. are just changed into different molecules with the help of maltase, sucrase and lactase. Maltose, sucrose, lactose, etc. are converted into the simple molecules or monosaccharides like galactose, fructose and glucose with the help of maltase, sucrase and lactase. So that is happening in the digestive system. Final products of carbohydrate digestion: 80% of glucose, 10% fructose and 10% galactose. Isosmotic absorption of isosmotic absorption of water. Water is transported through the intestinal membrane entirely by diffusion. Furthermore, this diffusion obeys the usual laws of osmosis. Therefore, when the chyme is diluted enough, water is absorbed through the intestinal mucosa into the blood of the villi almost entirely by osmosis. Brush border is there. Pinocytic vesicles can be seen, and endoplasmic reticulum, mitochondria. This indicates the brush border of gastrointestinal epithelial cells, showing also absorbed pinocytic vesicles, mitochondria and endoplasmic reticulum lying immediately beneath the brush border. Then therefore, when the chyme is dilute enough, water is absorbed through the intestinal mucosa into the blood of the villi almost entirely by osmosis. Conversely, water can also be transported in the opposite direction, from plasma into the chyme. This type of transport occurs especially when hyperosmotic solutions are discharged from the stomach into the duodenum. Within minutes, sufficient water usually will be transferred by osmosis to make the chyme isoosmotic with the plasma. The next one is the absorption of ions. Sodium is actively transported through the intestinal membrane. 20 to 30 grams of sodium is secreted in the intestinal secretions each day. In addition, the average person eats 5 to 8 grams of sodium each day. Therefore, to prevent net loss of sodium into the feces, the intestine must absorb 25 to 35 grams of sodium each day, which is equal to about 1/7 of all the sodium present in the body. Whenever significant amounts of intestinal secretions are lost to the exterior as an extreme diarrhea, the sodium reserves of the body can sometimes be depleted to lethal levels within hours. Normally, however, less than 0.5 of the intestinal sodium is lost in the feces each day because it is rapidly absorbed through the intestinal mucosa. So when we just try to look at the functional organization of villus, we can see there is portions called central lacteal region, blood capillaries and vein and arteries. And you just learn the parts of cross section showing a basement membrane beneath the epithelial cells and brush border. At the other end of the cell, we can see that brush border is there, basement membrane, venules are apart, then central lacteal, arteriole and capillaries are there. So the basic mechanism of sodium absorption from the intestine is very important. Sodium absorption is powered by active transport of sodium from inside the epithelial cells through the basal and lateral walls of the cells into the paracellular spaces. This active transport obeys usual laws of active transport. It requires energy, and the energy process is catalyzed by appropriate adenosine triphosphate enzymes in the cell membrane. Part of a sodium is absorbed along with chloride ions. In fact, the negatively charged chloride ions are mainly passively dragged by the positive electrical charges of the sodium ions. Active transport of sodium through the basolateral membrane of the cell reduces the sodium concentrations inside the cell to a low value. Because the sodium concentration in the chyme is normally about 142 equivalent per liter, about equal to that in plasma, sodium moves down this steep electrochemical gradient from the chyme through the brush border of the epithelial cell into the epithelial cell cytoplasm. Sodium is also co-transported through the brush border membrane by several specific carrier proteins, including sodium glucose co-transporter, sodium amino acid co-transporters, and the sodium hydrogen exchanger. Osmosis of the water. The next step in the transport process of osmosis of water by transcellular and paracellular pathways. This osmosis occurs because a large osmotic gradient has been created by the elevated concentration of ions in the paracellular space. Much of this osmosis occurs through the tight junctions between the apical borders of the epithelial cells, the paracellular pathway, but much also occurs through the cells themselves, the extracellular pathway. Osmotic movements of the water creates flow of fluid into and through the paracellular spaces and finally into the circulating blood of the villus. Aldosterone greatly enhances sodium absorption. When a person becomes dehydrated, large amounts of aldosterone are secreted by the cortex of the adrenal gland. Within 1 to 2 hours, this aldosterone causes increased activation of the enzyme and transport mechanisms for all aspects of sodium absorption by the intestinal epithelium. The increased sodium absorption in turn causes a secondary increase in absorption of chloride ions, water, and some other substances. This effect of aldosterone is typically important in the colon because it allows virtually no loss of sodium chloride in the feces and also little water loss. Thus, the function of aldosterone in the intestinal tract is the same as that achieved by the aldosterone in the renal tubules, which also serves to conserve sodium chloride and water in the body when a person becomes depleted of sodium chloride and dehydrated. Absorption of chloride ions in the small intestine. In the upper part of a small intestine, chloride ion absorption is rapid and occurs mainly by diffusion. Absorption of sodium ions through the epithelium creates electronegativity in the chyme and electropositivity in the paracellular spaces between the epithelial cells. Chloride ions then move along this electrical gradient to follow the sodium ions. Chloride is also absorbed across the brush border membrane of parts of the ileum and large intestine by a brush border membrane chloride bicarbonate exchanger. Chloride exists in cell on the basolateral membrane through chloride channels. Absorption of bicarbonate ions in the duodenum and jejunum. Often large quantities of bicarbonate ions must be reabsorbed from the upper small intestine because large amount of bicarbonate ions have been secreted in the duodenum in both pancreatic secretion and bile. The bicarbonate ion is absorbed in an indirect way as follows. When sodium ions are absorbed, moderate amount of hydrogen ions are secreted into the lumen of the gut in exchange for some of the sodium. These hydrogen ions in turn combine with the bicarbonate ions to form carbonic acid, which then dissociates to form water and carbon dioxide. The water remains as part of chyme in the intestine, but the carbon dioxide is readily absorbed into the blood and subsequently expired through the lungs. This process is also called active absorption of bicarbonate ions. It is the same mechanism that occurs in the tubules of the kidneys. Secretion of bicarbonate and absorption of chloride ions in the ileum and large intestine.
The epithelial cells on the surface of the villi of the ileum, as well as on all surface of the large intestine, have a specific capacity of secreting bicarbonate ions in exchange for absorption of chloride ions. This capability is important because it provides alkaline bicarbonate ions that neutralize acid products formed by bacteria in the large intestine. Active absorption of calcium ion, potassium, magnesium, and phosphate. Calcium ions are actively absorbed into the blood, especially from the duodenum region, and the amount of calcium ion absorption is exactly controlled to supply the daily need of food for calcium. An important factor controlling calcium absorption is parathyroid hormone secreted by the parathyroid glands, and another is vitamin D. Parathyroid hormone activates vitamin D, and the activated vitamin D in turn greatly enhances calcium absorption. These effects are very important. Ions are also actively absorbed from the small intestine. The principles of ion absorption and regulation of its absorption in proportion to the body's need for iron, especially for the formation of hemoglobin, is important. And potassium, magnesium, phosphate, and probably still other ions can also be actively absorbed through the intestinal mucosa. In general, the monovalent ions are absorbed in ease and in great quantities. Bivalent ions are normally absorbed in only small amounts. For example, maximum absorption of calcium ions is only 1 to 50 as great as the normal absorption of sodium ions. Fortunately, only small quantities of bivalent ions are normally required daily by the body. Absorption of carbohydrates. Carbohydrates are mainly absorbed as monosaccharides. Potassium, magnesium, phosphate, and probably still other ions are also actively absorbed. So essentially all the carbohydrates in food are absorbed in the form of monosaccharides. Only a small fraction is absorbed as disaccharides, and almost none is absorbed as larger carbohydrate compounds. By far the most abundant of the absorbed monosaccharides is glucose, which usually accounts for more than 80% of the carbohydrate calories absorbed. The reason for this high percentage is that glucose is the final digestion product of our most abundant carbohydrate food, the starches. The remaining 20% of absorbed monosaccharides is composed almost entirely of galactose and fructose, and galactose derived from milk, and fructose is one of the monosaccharide digested from cane sugar. Virtually all the monosaccharides are absorbed by a secondary active transport process, and we will first discuss the absorption of glucose. Glucose is transported by a sodium co-transport mechanism. In the absence of sodium transport through the intestinal membrane, virtually no glucose can be absorbed because glucose absorption occurs in a co-transport mode with active transport of sodium. The transport of sodium through the intestinal membrane occurs in two stages. First is active transport of sodium ions through the basolateral membranes of the intestinal epithelial cells into the intestinal fluid, thereby depleting sodium inside the intestinal epithelial cells. Second, a decrease of sodium inside the cells causes sodium from the intestinal lumen to move through the upper brush borders of the epithelial cells to the cell interior by a process called secondary active transport. Thus, the low concentration of sodium inside the cell literally drags sodium to the interior of the cell, and glucose is dragged along with it. Once inside the epithelial cell, other transport proteins and enzymes cause facilitated diffusion of the glucose through the cell's basolateral membrane into the paracellular space and from there to the blood. To summarize, it is the initial active transport of sodium through the basolateral membrane of the intestinal epithelial cells that provide the eventual force for moving glucose through the membranes as well. Absorption of other monosaccharides. Galactose is transported by almost exactly the same mechanism as glucose. Fructose transport does not occur by the sodium transport mechanism. Instead, fructose is transported by facilitated diffusion all the way through the intestinal epithelium and is not coupled with sodium transport. Much of the fructose on entering the cell becomes phosphorylated. It is then converted to glucose and finally transported in the form of glucose and rest of the way into the blood, because fructose is not co-transported with sodium. Its overall rate of transport is only about one half that of glucose or galactose. Now let's start the next topic, that is digestion and absorption of proteins. The learning objective include describe the steps involved in the breakdown and absorption of proteins. Major terms associated with this topic: amino acids, any group of organic compounds containing one or more amino group NH2 and one or more carboxyl groups COOH, they combine together to form proteins. Next, peptide linkage, amino acids bound together by the process of condensation, where a peptide linkage is formed when a hydroxyl ion is removed from one amino acid and the hydrogen is removed from the succeeding amino acid. Peptones, derivatives of proteins that are formed by partial hydrolysis of a protein. Polypeptides, peptides containing many amino acids, typically anything between 10 and 100 amino acids. Next, proteolytic enzymes, enzymes that help with hydrolytic breakdown of proteins into simple soluble substances as occurs in digestion. Then proteosis, a mixture of split products formed by the hydrolysis of proteins or polypeptides. Next, hydrolysis of proteins. Proteins are formed from multiple amino acids that are bound together by peptide linkages. At each linkage, a hydroxyl ion has been removed from one amino acid and the hydrogen ion has been removed from the succeeding one. Thus, the successive amino acids in the protein chain are also bound together by condensation, and digestion occurs by the reverse effect, hydrolysis. That is, the proteolytic enzymes return hydrogen and hydroxyl ions from water molecules to the protein molecules to split them into their constituent amino acids. Therefore, the chemistry of digestion is simply because in the same case of all three major types of food, the same basic process of hydrolysis is involved. The only difference lies in the types of enzymes required to promote the hydrolysis of reactions for each type of food. All the digestive enzymes are proteins. Then digestion of proteins. Proteins of the diet. Dietary proteins are chemically long chains of amino acids bound together by peptide linkages. A typical linkage is like OH and H are bonded. The characteristics of each protein are determined by the types of amino acids in the protein molecule and by the sequential arrangement of these amino acids. Then digestion of proteins in the stomach. Pepsin, an important peptic enzyme of the stomach, is most active at a pH of 2 to 3 and is inactive at a pH above about 5. Consequently, for this enzyme to cause digestion of proteins in stomach juices must be acidic. Gastric glands secrete a large quantity of hydrochloric acid. This hydrochloric acid is secreted by the parietal or oxyntic cells in the glands at a pH of about 0.8, but by the time it is mixed with the stomach contents and secretion from the non-oxyntic glandular cells of the stomach, the pH then averages around 2.0 to 3.0, a highly favorable range of acidity for pepsin activity. One of the important features of pepsin digestion is its ability to digest the protein collagen that is collected little by other digestive enzymes. Collagen is a major constituent of the intracellular connective tissue of meats. Therefore, for the digestive enzyme to penetrate meat and digest the other meat proteins, it is necessary that the collagen fibers are digested. Consequently, in persons who lack pepsin in the stomach juices, the ingested meats are less well penetrated by the other digestive enzymes and therefore may be poorly digested. A and B pepsin only initiates the process of protein digestion, usually providing only 10 to 20% of the total protein digestion to convert the protein to proteoses, peptones, and few polypeptides. The splitting of proteins occurs as a result of hydrolysis at the peptide linkage between amino acids. Most protein digestion results from actions of pancreatic proteolytic enzymes. Most protein digestion occurs in the upper small intestine, in the duodenum and jejunum, under the influence of proteolytic enzymes from pancreatic secretion. Immediately on entering the small intestine from the stomach, the partial breakdown products of the protein foods are attacked by the major proteolytic pancreatic enzyme trypsin, chymotrypsin, carboxypolypeptidase, and elastase. Then both trypsin and chymotrypsin split protein molecules into small polypeptides, carboxypolypeptidase, then cleaves individual amino acids from the carboxyl ends of the polypeptides. Proelastase in turn is converted into elastase, which then digests elastin fibers that partially hold meats together. Only a small percentage of the proteins are digested all the way to their constituent amino acids by the pancreatic juice. Most remain as dipeptides and tripeptides in the mouth. That is happening. There is an enzyme, no enzymes to digest protein. Next is the esophagus region. There is also no enzymes. After that, stomach. There is pepsin. So that pepsin converts proteins to proteoses, peptones, and polypeptides. 10 to 20% of protein digestion is happening there. Next stage is in the small intestine. Small intestine has many enzymes like trypsin, chymotrypsin, carboxypolypeptidase, proelastase. They just convert protein into proteolases, peptones, polypeptides plus amino acids. Then aminopolypeptidases and dipeptidases are there. They just convert this polypeptides into tripeptide, dipeptide, and amino acids. Then enterocytes are there within cytosol. They just contain multiple peptidases enzymes. Then they just convert dipeptides and tripeptides into amino acids. So this is what is happening in the digestive system in case of protein digestion. So digestion of peptides by peptidases in the enterocytes that line the small intestinal villi. The last digestive stage of a protein is the intestinal lumen is achieved by the enterocytes that line the villi of the small intestine, mainly in the duodenum and jejunum. The cells have a brush border that consists of hundreds of microvilli projecting from the surface of each cell. In the membrane of each of these microvilli are multiple peptidases that protrude through the membranes to the exterior, where they come in close contact with the intestinal fluids. Two types of peptidase enzymes are especially important: aminopolypeptidases and several dipeptidases. They split the remaining larger polypeptides into tripeptides and dipeptides, and a few into amino acids. The amino acids, dipeptides, and tripeptides are easily transported through the microvillar membrane to the interior of the enterocyte. Finally, inside the cytosol of the enterocyte are multiple other peptidases that are specific for the remaining types of linkages between amino acids. Within minutes, virtually all the last dipeptidases and tripeptidases are digested to the final stage to form single amino acids, which then passes through the other side of enterocyte and hence to the blood. More than 99% of the final protein digestion products that are absorbed are individual amino acids, with only rare absorption of peptides and very rare absorption of whole protein molecules. Even when few absorbed molecules of whole protein can sometimes be seriously allergic or immunological disturbance can be produced. Basic principles of gastrointestinal absorption. The basic principles of gastrointestinal absorption and absorption of proteins is dipeptides, tripeptides, and amino acids. As explained earlier, most proteins after digestion are absorbed through the luminal membranes of the intestinal epithelial cells in the form of dipeptides, tripeptides, and a few free amino acids. The energy for most of this transport is supplied by a sodium cotransport mechanism in the same way that sodium cotransport of glucose occurs. That is, most peptide or amino acid molecules bind in the cell's microvillus membrane with a specific transport protein that requires sodium binding before transport can occur. After binding, the sodium ion then moves down its electrochemical gradient to the interior of the cell and pulls the amino acids or peptide along with it. This process is called co-transport or secondary active transport of the amino acids and peptides. A few amino acids do not require the sodium co-transport mechanism, but instead are transported by special membrane transport proteins in the same way that fructose is transported by facilitated diffusion. At least 5 types of transport proteins for amino acids and peptides have been found in the luminal membranes of intestinal epithelial cells. This multiplicity of transport proteins is required because of diverse binding properties of different amino acids and peptides. Next is the digestion and absorption of fats. Describe the steps involved in the breakdown and absorption of fats. Major terms associated with this topic are emulsification, the process of breaking up large fat globules into small globules with the help of bile salts, and lecithin, and agitation. Then lecithin, a class of phospholipid containing glycerol, phosphate, choline, and fatty acids. They help in the emulsification of fats during the process of fat digestion. Then micelles, small spherical cylindrical globules 3 to 6 nm in diameter, composed of 20 to 40 molecules of bile salts, and help in the digestion and absorption of fats. Then triglycerides, the combination of 3 fatty acid molecules condensed with a single glycerol molecule. Digestion of various foods by hydrolysis of fats. Almost the entire fat portion of the diet consists of triglycerides or neutral fats, which are combinations of 3 fatty acid molecules condensed with a single glycerol molecule. During condensation, 3 molecules of water are removed. Hydrolysis or digestion of the triglycerides consists of the reverse process. The fat digesting enzymes return 3 molecules of water to the triglyceride molecule and thereby split the fatty acid molecules away from the glycerol. So digestion of fats of the diet. By far the most abundant fats of the diet are the neutral fats, also known as triglycerides, each molecule of which is composed of glycerol nucleus and 3 fatty acid side chains. Neutral fat is a major constituent of food of animal origin, but much less so in food of plant origin. Small quantities of phospholipids, cholesterol, and cholesterol esters are also present in the usual diet. The phospholipids and cholesterol esters contain fatty acid and therefore can be considered fats. Cholesterol is a sterol compound that contains no fatty acids, but it does exhibit some of the physical and chemical characteristics of fats. In addition, it is derived from fats and is metabolized similarly to fats. Therefore, cholesterol is considered from a dietary point of view to be fat. Digestion of fats occurs mainly in the small intestine. A small amount of triglyceride is digested in the stomach by lingual lipase secreted by lingual glands in the mouth and swallowed in the saliva. The stomach also secretes a small amount of gastric lipase from the fundic region. This amount of digestion is less than 10%, as is generally unimportant. Instead, essentially all fat digestion occurs in the small intestine. Steps in fat digestion. Emulsification by bile acids and lecithin. The first step in fat digestion is to physically break the fat globules into small sizes so that the water-soluble digestive enzymes can act on the globule surface. This process is called emulsification of the fat, and it begins with agitation in the stomach to mix the fat with the products of stomach digestion. Most of the emulsification then occurs in the duodenum under the influence of bile, which contains large quantities of bile salts as well as the phospholipid lecithin. Both of these substrates and substances, but especially the lecithin, are extremely important for emulsification of the fat. The polar parts, the points where localized ionization occurs in water of the bile salts and lecithin molecules, are highly soluble in water, whereas most of the remaining portion of their molecules are highly soluble in fat. Therefore, the fat-soluble portions of this liver secretion is dissolved in the surface layer of the fat globules with the polar portions projecting. The polar projections in turn are soluble in the surrounding watery fluids, which greatly decrease the interfacial tension of the fat and makes it soluble as well. When the interfacial tension of a globule of non-miscible fluid is low, this non-miscible fluid on agitation can be broken up into many tiny particles far more easily than it can when the interfacial tension is great. Consequently, a major function of the bile salts and lecithin, especially the lecithin in the bile, is to make the fat globules readily fragmentable by agitation with the water in the small bowel. This action is same as that of many detergents that are widely used in household cleaners for removing grease. Because the average diameter of the fat particles in the intestine after emulsification has occurred is less than 1 μm, this represents an increase as much as 100-fold in total surface area of the fats caused by the emulsification process. So what is happening inside our body to help the digestion and absorption of fats? In the mouth, there is an enzyme called lingual lipase. Lingual glands of the mouth secrete them, that lingual lipase. This leads to the digestion of about less than 10% of digestion of triglycerides. Then esophagus, stomach contain enzymes called gastric lipases. Fundus area of the stomach produce them. They lead to about less than 10% of the total digestion of triglycerides again. Then stomach contain this gastric lipase, which is produced by the fundus area of the stomach. Never forget that. Then small intestine is there, large intestine is there. Small intestine contain enzymes called pancreatic lipases. Essentially, all triglycerides are digested by pancreatic lipase to monoglycerides and glycerol. Then enteric lipase is also there. Enterocytes lining the villi produce them. So they act no significant action by them, as pancreatic lipase digests most triglycerides. So the lipase enzymes are all water-soluble compounds and can attack the fat globules only on their surface. Consequently, this detergent function of bile salts and lecithin is very important for digestion of fats. Triglycerides are digested by pancreatic lipase. By far the most important enzyme of digestion of the triglyceride is pancreatic lipase, present in enormous quantities in pancreatic juice, enough to digest within 1 minute all triglycerides that it can reach. The enterocytes of the small intestine contain additional lipase known as enteric lipase, but it is usually not needed. End products of fat digestion are free fatty acids. Most of the triglycerides of the diet are split by pancreatic lipase into 3 fatty acids and 2 monoglycerides, bile salts from micelles that accelerate fat digestion. The hydrolysis of triglycerides is a highly reversible process. Therefore, accumulation of monoglycerides and free fatty acids in the vicinity of digesting fats quickly blocks further digestion. However, the bile salts play the additional important role of removing the monosaccharides and free fatty acids from the vicinity of the digesting fat globules almost as rapidly as these end products of digestion are formed. This process occurs in the following way. When bile salts are of a high enough concentration in water, they have the propensity to form micelles, which are small spherical cylindrical globules 3 to 6 nm in diameter, composed of 20 to 40 molecules of bile salt. These micelles develop because each bile salt molecule is composed of a sterol nucleus that is highly fat soluble and a polar group that is highly water soluble. The sterol nucleus encompasses the fat digestate, forming a small fat globule in the middle of a resulting micelle, with polar groups of bile salts projecting outward to cover the surface of the micelle. Because these polar groups are negatively charged, they allow the entire micelles globule to dissolve in the water of the digestive fluid to remain in stable solution until the fat is absorbed into the blood. The bile salt micelles also act as a transport medium to carry the monoglycerides and free fatty acids, both of which would otherwise be relatively insoluble to brush borders of the intestinal epithelial cells, where they are absorbed into the blood. Digestion of cholesterol esters and phospholipids. Most cholesterol in the diet is in the form of cholesterol esters, which are combinations of free cholesterol and one molecule of fatty acid. Phospholipids also contain fatty acids within their molecules. Both the cholesterol esters and the phospholipids are hydrolyzed by two other lipases in the pancreatic secretion that free the fatty acids. The enzyme cholesterol ester hydrolase to hydrolyze the cholesterol ester and phospholipase A2 by hydrolyzing the phospholipid. The bile salt micelles play the same role in ferrying free cholesterol and phospholipid molecules digests that they play in ferrying monoglycerides and free fatty acids. Instead, essentially, no cholesterol is absorbed without this function of micelle. Absorption in the small intestine. Absorption of fats. Earlier in this chapter, it was pointed out that when fats are digested to form monosaccharides, the free fatty acids, both of these digestive end products, first become dissolved in the central lipid portion of bile micelles, because the molecular dimensions of these micelles are only 3 to 6 nm in diameter, and because of their highly charged exterior, they are soluble in chyme. In this form, the monoglycerides and free fatty acids are carried to the surface of the microvilli of the intestinal cell brush border and then penetrate into the recess among the moving, agitating microvilli. Hence, both the monosaccharides and monoglycerides and fatty acids diffuse immediately out of the micelles and into the interior of the epithelial cells, which is possible because the lipids are also soluble in the epithelial cell membrane. This process leaves the bile micelles still in the chyme, where they function again and again and help absorb still more monoglycerides and fatty acids. Thus, the micelles perform a ferrying function that is highly important for fat absorption. In the presence of an abundance of bile micelles, about 97% of the fat is absorbed. In the absence of the bile micelles, about 40 to 50% can be absorbed. After entering the epithelial cell, the fatty acids and monoglycerides are taken up by the cell's smooth endoplasmic reticulum. Here they are mainly used to form new triglycerides that are subsequently released to form chylomicrons through the base of the epithelial cell to flow upward enough the thoracic lymph duct and empty into the circulating blood. The direct absorption of fatty acids into the portal blood. Small quantities of short and medium chain fatty acids, such as those from butter fat, are absorbed directly into the portal blood rather than being converted into triglycerides and absorbed by way of the lymphatics. The cause of this difference between short and long chain fatty acid absorption is that the short chain fatty acids are more water soluble and mostly are not reconverted into the triglycerides by the endoplasmic reticulum. This phenomenon allows direct diffusion of this short chain fatty acids from the intestinal epithelial cells directly into the capillary blood of the intestinal villi.